Se inicia sesión.
Session starts. Good morning, everybody. It's a pleasure to greet you today. I'm Carla Sáenz, Regional Advisor of Bioethics at PAHO. Before starting the session, I will explain some logistical aspects. First, there will be presentations in English. Those who want to listen to the Spanish interpretation, go to the lower part of your screen and click on the interpretation globe and click on Spanish, so as to be able to listen to the Spanish interpretation of the presentations and the session.
This is a bilingual session. So if you want to listen in English to all the sessions, please go to the bottom the bottom tab of the Zoom. There's a little globe that says interpretation, and click on English. Then that way you will hear everything in English. And hope that works for everybody. It's great to be with you today. Bienvenidos a una nueva sesión de nuestra serie de seminarios virtuales.
Welcome to a new session of our virtual seminars on clinical trials. This is a joint effort between the Bioethics Regional Program in the Evidence and Health Evidence at PAHO and the Quality Unit and Medicines Regulation of PAHO. That is part of our Department of Innovation, Access to Medicines and Health Technologies. Before starting, if you have any questions, you can send them in Spanish, in English, or in Portuguese through the Q&A function. Any other comments, if you want to share any links, you can do so in the chat. But please, your questions and comments, it's better to have them in the Q&A button because it allows us to better identify the question. We will not give an attendance certificate for this seminar. The session will be recorded and will be shared as we shared the previous seminars through our channels. The list of ethics research of PAHO, the website of the Bioethics Regional Program of PAHO, and our America's Clinical Trials portal that I invite you to visit, because we have also other resources, recent resources. For example, our model sample contract for clinical tests that is already also available in English. And it is through that list that we will invite you to participate in the next webinar on the revised concept of vulnerability and its significance for clinical tests. That will take place on November 7th in the afternoon. Today is clinical tests or essays on advanced therapies, focusing on technical and regulatory aspects. There's an ethical review and a regulatory vigilance. Ethical standards for research are applicable for all research. Each research focus on the same possible benefits, risks, etc., etc. However, I want to stress that even if the focus is in the scientific and regulatory aspects of clinical trials. But this is also very important for ethics committees that need to understand the underlying science and what it implies to do clinical trials in our region for a rigorous research in the topic. We will start today's session talking about the regulation of advanced therapies in the European Union. Specifically, what are the advanced therapies and how is this regulated in Europe through the EMA throughout the product life cycle? And we have the pleasure to have for that session Dr. Marcos Timon, Head of Unit Biological Products, Advanced Therapies and Biotechnology at the Spanish Agency for Medicines and Health Products. He's also a member of the Advanced Committee and he worked as a researcher before devoting his work to the regulatory area. Dr. Timon, please take the floor. Thank you very much, Carla, for your introduction, for the invitation to participate. Yeah, I was trying to turn on my camera. I'll try to share my presentation. I hope you can see it well. Can you see my presentation? Yes, we do. Thank you very much for the invitation. Good morning to most of you. Good afternoon to some of you, some of us who are on the other shore of the ocean. I work in the European Medicines, the Spanish Agency for Medicines, part of the EMA, European Medicines Agency. The concept of advanced therapy is of a purely regulatory nature. If I understand well, this was invented precisely in Europe. There's no This is simply a regulatory term. It doesn't imply better or worse quality. Most innovative products belong to this category. Advanced therapies in the European Union are regulated in this legal regulation 1394/2007. There are different legal document categories in the European Union. Regulations are a type of document that do not require to be transposed to the national laws of each member state. It simply goes into force in all member states. This is very important. In advanced therapies, all member states of the EU have the same legal document, and the only thing that is done is to translate the text to any pertinent language, but the content is exactly the same. This legal text entered into force at the end of 2008, so we have quite a significant experience working with the document. And what it defined as advanced therapies are these three general categories of medicines: gene therapy, somatic cell therapy, and tissue engineering, but there are also requirements and adaptations with the general medicines regulations that was necessary because these are very particular types of medicines. Obviously, we need to define these subcategories. These are the definitions in the legal text. This is the legal jargon, which is quite complicated. Well, gene therapy, medicinal products defined in the regulation, because there was no previous regulation in the EU. But gene therapy and somatic cell therapy had prior definitions, but when the regulations were published, that definition was updated and adjusted to the scientific evolution. So the new definitions that are duly adapted and were published in Directive 2009-120. I will address each of these categories, addressing their different definitions. Gene therapy, the legal definition says that it's a biological medicine. This is slightly different from chemical synthesis products. In Europe, gene therapy is only applied in Europe for the time being to biological medicines with the following characteristics. First, that it has to contain a recombinant nucleic acid. It, it has to be obtained not naturally but through genetic engineering or gene engineering technology. And the second requirement is that its therapeutic effect is directly related with the nucleic acid sequence or with the product of that sequence's expression. This excludes medicines these gene therapy medicines don't include infectious disease vaccines, not through scientific meaning. This is purely regulatory in nature because vaccines were already well regulated and their clinical trials are very different from the other therapeutic indications that include gene therapies. So this is a regulatory definition. There are vaccines for COVID-19, for example, based off an mRNA. But in Europe, this is outside the definition of advanced therapy. I repeat, this is for infectious disease vaccines, which are excluded from this regulation. There are different types of gene therapy based on different themes, both in vivo gene therapy, where the nucleic acid is extracted from the patient, and also ex vivo approaches, where cells are modified outside of the patient and then they are injected into the patient. All of this in Europe is defined as gene therapy, and also there are manifold vectors. You know that the nucleic acid is very difficult to obtain, so we use different types of vectors. The most common are those in this table, but they are not the only ones. Most of vectors are based on viruses, which are good tools to transfer genetic material. All depends on what we want to obtain, the size of the nucleic acid sample, etc. And there are also non-viral vectors, such as lipid nanoparticles that are used mostly to transfer mRNA. As to somatic cell therapy medicinal products, we can group them in cell-based medicines, which are very similar from others and are only distinguished by the mechanism with which they are obtained. I won't address the complete definition, which is in the regulatory documents that I have mentioned, but a summary or the most important of the criteria used to decide whether a medicine is somatic or not, of cells or tissues. These are medicines that contain cells or tissues regardless whether the final product is the somatic cellular therapy or tissues, and the one of the both requirements have to be complied with. The first requirement is that cells or tissues must have been submitted to substantial manipulation, or that the cells and tissues have an essential function which is different in the donor and the receptor, the receiver. This is interpreted in a wide sector. It can be the same patient where we take a sample as a donor and then we administer it as a receiver if the essential function is different in the donor and the receiver. We used We use tissue engineering or cell therapy that makes it possible to regenerate, restore, or replace a human tissue. Then it's tissue engineering. And then if the action is pharmacological, immunological, or metabolic, the cell, we are in the presence of somatic cell therapy. These cells can be stem cells, whatever the type. Basically, two types: trunk cells or stem cells in English. And what distinguish stem cell are these two characteristics. One, that they can divide and make copies of themselves, and two, that in specific circumstances they can differentiate into many cell types. And here there are several types of stem cells: embryonic stem cells, iPS cells induced, or adult stem cells. Each one of these cells have a different pluripotentiality. Full stem cells can be differentiated into any types of cells in the body, and non-embryonic origin. And this involves an ethical factor. When it comes to embryonic stem cells, and then adult stem cells have a smaller capacity to differentiate it depending on the tissue. All these cells can be used to generate cell-based medicines or advanced therapy medicines. And of course also we can use completely differentiated cells, simply generating a larger number of these cells by identifying them some way before proceeding with the process. Advanced therapy medicines, as any other medicines, need to demonstrate their safety and effectiveness profile in clinical trials and the regulatory aspects. And at the end of the clinical development, there is a dossier to try to obtain a marketing authorization that makes it possible to administer this medication or this medicine to all patients after the experimental phase. There are different ways in Europe to authorize these procedures. National ways, so you can apply for an authorization, and each study member state national authorizations or other recognitions managed by the EMA by law. Advanced therapies in Europe have to go through procedures coordinated by EMA. This is the centralized procedure. All the procedure is coordinated by the EMA, the European Medicines Agency, and also initial assessment performed by two national competent authorities. If the EMA issues an authorization, the applicant can access to all EU markets. But there's one single authorization, but there are also some responsibilities at the purely national level, which is the authorization for clinical trials, which is within the purview of each member state where those tests will be conducted, and then the authorization and the decision whether to reimburse the medicine through the public system in all EU members. And then there's also the involvement of the company that conducted the trials. But the regulation left a door open, Article 28, establishing that in certain specific conditions, there can be an authorization to market. So through the national area, not needing to go through the European Union. This is generally applied and restricted to very specific circumstances where a medicine will be used and individually for a specific patient under the responsibility of a supervisory doctor, and this is produced in a non-routine basis. This is a bit old definition that has allowed each member state to implement this differently. Maybe this is not optimal because now we have different interpretations about this Article 28. In Spain, we interpreted and implemented it in our national laws. We did that in 2014, and these are our regulations in Spain. It was decided that the medicines through this way would not receive marketing authorization as such, but what we determined to be authorization for use for a specific hospital, which would be the applicant. So the medicine can only be used within the authorized hospital institution, even though the manufacturing can be outsourced outside of the hospital. Authorizations will be published in our webpage. They are valid for 3 years and can be renewed. Hospital exemptions in Spain forces the recipient hospital to assume certain responsibilities, for example, to assure the traceability, to conduct pharmacovigilance, and report annually to the agency about the uses, and of course ensure the quality, safety, and ethics and effectiveness. As for other authorized ATMPs, this is similar to what has been done in other countries, and here we decided to in this exemption to exclude to include the experimental medicines. We decided to include these experimental products to test their
Otra
safety when
característica de estas
we have
denominadas es su
tested the
complejidad en
safety.
comparación con cualquier otra
Human
categoría de
medicinal products,
medicamentos y
different
eso ha hecho
levels of
que
complexity
hayamos
as any
tenido que
other
adaptar
medicine,
muchos de
so
los
we
requisitos
have
generales
to
de otros
had
medicamentos
to adapt
para
many of
poder
the general
conseguir
requirements
que
of other medicines
estos medicamentos tan complejos puedan cumplir,
to
¿no?
make
Entre otras
sure that
adaptaciones, se ha
these complex
elaborado unas
medicines can
guías de
meet the
correcta fabricación,
requirements.
normas de correcta fabricación, de GMPs, específicas para
This is
aplicación
the
terapia
rules
avanzada en salud
governing
y
the
con
medicinal
ciertas
products in the
flexibilidades
EU,
para que
Volume 4
eh,
GMPs, Good
puedan
Manufacturing
cumplir
Practice.
las normas y
This
por tanto
ensures that
entrar
standards are
por la vía
duly
legal.
complied with in all the EU.
Vale. Como les dije, la
As I
clasificación de
said,
medicamentos
the
basados en células consiste en estos
classification
dos
of
requisitos y no
cell-based
siempre es fácil
medicines
decidir qué
are
consideramos una
consistent
manipulación
with
sustancial,
these
ya
two
que todos aquellos
requirements.
investigadores que hayan
Cells or
trabajado
tissues
con células pues saben que
subjected to substantial
cualquier
manipulation. This is
afinidad
not
que se
easy
haga
to
con las células,
define.
una simple centrifugación, pues
So it
seguramente
could be just
cambia
a process for
muchas de las características de la
centrifugation,
célula.
and this
Pero
could
aquí estamos
change the
hablando de
characteristics of the cell. But here
actividades
we are referring to
que
activities
constituyan una
that
manipulación
are
sustancial y eso hay que
substantial
interpretarlo. Y lo
manipulation, and
mismo
this
también
has
a
to be
veces por
interpreted.
interpretar
And the
que
same applies to
consideramos una función
what
esencial distinta entre
we
donante y el receptor.
consider an
Entonces,
essential
si les sirve
function between the donor
de
and the
orientación,
receptor.
el CAT, el
So,
Comité de Terapias
as
Avanzadas,
a
elaboró
guideline,
un
the
documento que es el
Committee on
técnico aquí, donde
Advanced
se dan algunas ideas
Therapies
de que
has developed
interpretamos o cómo
this
interpretamos
document with some ideas as to how to
estos criterios,
interpret
¿no?
these
Y también
criteria.
hay una lista
And there is
en este
also a
link
list
en la EMA
as
donde
part
pueden
of
ver
this link
clasificaciones de productos
from
que han
EMA, where
venido
you can see the
voluntariamente a
classification
ser
of
clasificados
drugs that
en
have
la EMA
been
y los criterios se
certified
han seguido
within
para
EMA and the
clasificarlos como
criteria
medicamento
followed
de
to
terapia avanzada
classify
o
them
no.
as
Voy a ir
advanced
terminando. Esta es
therapy drugs or not.
la lista más
To
actualizada que
conclude,
tenemos de
this
medicamentos
is an
autorizados
updated
por el
list of
procedimiento centralizado en
drugs
la
that
EMA,
we
terapias
have
avanzadas hasta ahora. Hemos
approved through
dado
EMA through
30
the
autorizaciones,
centralized procedure.
probablemente no
We have
sean
granted 30
demasiadas en comparación con
authorizations. They're
otros
not too
grupos
many
terapéuticos,
as
pero
compared to
como
other
digo,
therapeutical groups, but
son
as I
medicamentos muy
mentioned
complejos
before,
y a
these are
veces
quite
es
complex
difícil demostrar
medicines, and
seguridad y
oftentimes
eficacia.
the idea is
Además de
to
estos
prove
30,
safety and
8 han
efficacy.
sido
In addition
retirados
to
ya
these
del
30,
mercado después de
8 have
autorizarlos.
been withdrawn
Tengo
from the
que
market
matizar que
upon
todos
approval, and I
los
should
medicamentos que se han
add
retirado han sido
that all of
por
the
motivos de
documents withdrawn from the market,
marketing o
removed
por
from the
motivos de
market,
negocio de
had
la
to do
compañía.
because of
Ningún
marketing
nunca se
reasons or
retiraron medicamentos
business
de
reasons of the
terapias
company,
avanzadas por problemas de
and
seguridad.
none had any safety
Em,
considerations.
la mayoría como pueden ver aquí, de los medicamentos
As you can
autorizados son
see,
de terapia
most of
génica.
the drugs
Obviamente
that
tenemos
were
muy poco de
approved
terapia celular somática
are
y de
based on
ingeniería de tejidos.
gene
Y por lo que
engineering,
percibimos
gene
en otras
therapy,
actividades, por
but
ejemplo,
not
ensayos clínicos o asesorías
tissue
científicas,
engineering. And
digamos que la
we
mayoría de la
clearly
actividad ahora mismo
provide technical
está en el ámbito
advice, and most
de
of
la
it
terapia
is
génica.
within the area of gene
Esta es una
therapy.
actualización porque
This
la tabla
is
anterior se
updated
publicó hace unos
information.
meses.
The
Desde entonces se ha
previous table
autorizado
was
una
published a
nueva
couple of
terapia
months
celular,
ago, and this is the
Cell-Pro.
approval of a
Se ha
new
rechazado
cell
una solicitud de
therapy,
Levidis, que es una
Zemcelpro.
terapia génica para
It is
distrofia
a
muscular de Duchenne, y dos
request
solicitudes
for
han sido retiradas por parte de
a
los
muscle
solicitantes
dystrophy
cuando estaban ya al final de la evaluación.
medicine was
Uno es
rejected.
Amtagvi, que son
That
unos
was Elvidis.
linfocitos
And
infiltrantes de
then we
tumor
have
para el
two that
melanoma,
were
y el otro
withdrawn,
es Fanskia,
Amtagvi, that
que
is
es una terapia génica para
TIL
la
for
anemia de
advanced
Fanconi.
melanoma, Efansika, that is for
Aparte
the
de los
sickle
30
cell
autorizados por vía
anemia.
centralizada, tenemos
And
5
then,
autorizados
in
por
addition to
la
the
sola
ones that
inclusión
were
hospitalaria
authorized from the
en España,
centralized
y esto
fashion,
incluye a 2
we have
medicamentos basados
others
en
that
células CAR-T que
include
también tenemos autorizados por vía nacional. Realmente los
that include
medicamentos
drugs that were
basados en
authorized
células CAR-T
nationally.
han
The
supuesto una
document,
revolución en el
the
ámbito de las terapias avanzadas desde
the
los dos
medicines
primeros
that are
que
CAR
la
T
FDA
cells
autorizó en el
based
año 2017 y
were
Europa, la EMA, en el año
approved in
2018.
2017 and
Hasta ahora
2018
tenemos
by
7
EMA.
medicamentos
We
ya
already
autorizados
have
por la EMA y 2 más
seven
autorizados en
drugs,
España
7 medicines
por
that
vía
were
nacional.
approved by EMA,
Otro
and
grupo de
others that were
medicamentos de
approved
terapia
nationally.
avanzada donde está
Other
habiendo muchísimos
advanced
desarrollos es aquellos que
medicinal
utilizan
products are the ones
como
that
vector de administración un
that
virus
we use for
adenosizado.
administration in the case
Esto es
of
así porque
associated adenoviruses. The AAV, in its natural form, is quite safe and it is present in most individuals and animals. They are not pathogenic. They are not able to replicate by itself. The natural form is quite safe, and the modified form that we use to as the messenger for the DNA is quite safe. We have 7 products that are based on these products, and this includes gene therapy for hemophilia A, B, and also spine dystrophy. There has been quite a great deal of development in the use of first gene editing therapy, and we have had an authorization also the first medicine based on this technology, that is Cancevi. I am not going to offer any more details, but there are several technologies available for editing the human genome, and this is different from the typical approaches on gene therapy that had to do with the addition of a corrected version of the gene. Here, the idea is to interfere with the sequence and to modify the strand so that it is not considered complete. And then the mechanism of action of this medicine that has been authorized is Casgevy, and that is to inhibit an inhibitor to have the gamma globin expressed and also to correct 2 indications, the beta thalassemia being one of them that has a problem in the strand within the beta globin. So when we correct that hemoglobin, the persons have the right phenotype, and then the gamma globin can be properly expressed and the effect can be corrected. Within EMA, we have also organized a gene therapy, gene editing workshop, and this is the link that has the full video of the event. I also wanted to underscore this other medicine, Bioethics, for that it is administered directly to the skin to suppress the defective gene that is not properly expressed in these patients. What are the difficulties that we face with these medicines? The most important has to be the cost. These are very expensive medicines, and in particular, these medicines have the problem of the cost. This is something that we need to face. Another problem is the unregulated use. That's why it is relevant, and I wanted to underscore that the member states that have decided to regulate them have made the right decision, even in some areas of the world where this is deeply regulated. We spend a great deal of time to try to find areas or clinics that offer illegal treatments. We are trying to identify them to remove them from the market. So it is better to offer this so as to have therapies that are already tested, approved, and that are safe without endangering the life of the patient. And these are the signs that we see even at the hairdresser offering stem cell treatment. Thank you very much, and I will be available to answer any question that you may have at the end of the session. Thank you very much. It has been an extremely clear and illustrative session. I think that we have all seen that unproven interventions are usually offered, but I had never seen it at the hairdressers. Questions will be asked later, but now I would like to hear the researcher. What are the challenges in research with advanced therapies? And to that end, we have Gustavo Sebleler, who is a doctor, director of research and teaching at FLENI, and the director of the INEU, FLENI CONICET Argentina. He is a prolific researcher in pathology, and his research covers dementia, biomarkers, brain, the biology of stem cells, genome, etc. It is a pleasure to have you here with us. Gustavo, please go ahead. Hello. I don't know if you can hear me. Yes. And we can see you perfectly well. Thank you. First, I'd like to thank the invitation to the bioethics group within PAHO and also to the other participants. It is a pleasure to discuss with you a topic that, in my opinion, is a metaphor of the advance made in biomedics, what we learned with stem cells, and this is something that I will be referring to now. This is something that can be applied to many other areas that have to do with innovation and also the production of biomedical knowledge that can be transferred to doctors. This is a 1998 paper from the University of Wisconsin by James Thompson, who published that the human blastocysts, when they are used with specific culture liquids, could mature to any adult liquid. This was later proved by the Thompson group, and when we thought in biology that there was a fate for the cells, a manifest destiny for those cells, the type of culture, the culture was the one that was going to be the final use, the final fate for that cell. And we see that these cell lines may be used in human developmental biology, drug discoveries, and transplantation medicine. And later on, this is prior work, but both were combined, and this is the famous sheep dory, the cloned mammal that led to human cloning and the use of these technologies. There were some ethical issues, and I don't think that they were very positive for the field back then. Basically, we have three comments: the biological phenomenon, the impact, and also the clinical use. And we're going to see then the impact it has on society and the media. The biological phenomenon is spectacular, and back then it was even more spectacular. It is a cell that back then had the embryos that did not get implanted as part of the in vitro fertilization process. A large number of embryos was needed because the technique was not very efficient. So those embryos that the couple decided were not going to be used because they already had one or two babies. This is the basis for Thompson's work, where he showed that the pluripotent, the unipotent cells can be used based on the culture media. This was already seen, but just to review the embryonic cells, that is the beginning of the theories on stem cells from embryos that led to a highly complicated issue in some societies. You would recall the definition by President Bush Jr., who referred to the prohibition of using stem cells that originated from an embryo, and also the induced pluripotent stem cells that was communicated by Yamanaka that led to the Nobel Prize a couple of years ago in the somatic stem cells derived from the umbilical cord, the blood, and the non-hepatogenetic one from the bone marrow. These are the three large groups of stem cells that we are familiar with. And the great contribution that we had with Yamanaka was the description of the gene editing technique that is much more efficient than the one that we had back then, and that allowed us to operate in a simpler and more previsible manner, and also the Nobel Prize to chemistry to the two individuals, to the two scientists. But what was the impact it had on the media? Because this was something that conditioned the development of biomedicine and stem cells. We have 3 publications, the New York Times, Time Magazine, and a national publication in Argentina, Noticias, where it says the cure for all diseases, the revolution of stem cells can save your life. So there is an expectation that was created that was impossible to meet, and society started to think that stem cells were going to cure us of all diseases, and we also saw it in cosmetic sector, that is to use the use of stem cells to sell dermatological lotion, and then we heard about the reparative medicine. Instead of being centered on pharmaceuticals, one could go to sort of a shop and ask for a femur or a liver or whatever it would be to replace the one that was sick. That was the result of the impact it had in the media. And in the case of Argentina, there was a significant influence of the Chinese clinics. They were not the only ones, but they clearly promised this. Here, you can see there was no statistics, just individual experience of patients, and they cured so many things that there was a menu where they could see Alzheimer and facial paralysis, and clearly there was someone that was about to die and then would undertake marathons, or you would become a high-performing athlete. It was like a panacea. That's why I have that goddess there. It seemed to be the promise to cure all the diseases, and clearly, in a way, this was taken to its limits. They wouldn't cure what they said that they were going to cure. They charged a fortune. There was no regulation. There were conflicts with the regulatory authorities. They ended up with justice in the Justice Department. So this complicated the issue of the stem cells. And let's look at the medical use. We have been working with iPS cells, and this is the model of diseases, in particular of the central nervous system. If we look at this, for some years, we have had a classification of diseases according to the mechanics, the relevance for individuals and production. And these cells combine basically all of them, including the autopsy tissue, that is the final film, more than the animal models that are not so important, or non-human primates. Now we have the model of a disease in an in vitro situation that is endless. And let me share with you some of the experiences that we had in our lab. We were lucky to identify, as part of a consortia, a new mutation of PSEN1. It was a mutation in an Argentine family, and this is the genealogic tree. This is a mutation that was not described, and starting with that mutation that was obtained with bioinformatics, we were able to generate cells through the Yamanaka technology. This cell is known as FFAD1, and repositories identified here, and in culture that cell produce organoids. So the mutation was in all of the cells, including the somatic cells, and in this case, this was a skin biopsy, and we were able to obtain a model of the Alzheimer's disease in vitro based on human cells so these reproduce the proteins that are typical of the Alzheimer's disease. This is a model that was later on expanded and used with other diseases. Here we see a very rare disease with a missense mutation and linked to familial muscular dystrophy. And these models allowed to prove in vitro models, and also in the case of the linkage here to dementia and the ORF mutation, we identified the mutation that are characterized with the hereditary variants, and oftentimes this was also overlapping with dementia, and we can see how this starts to express the phenotype of the in vitro disease. Also, there was a myofibrillar myopathy, and recently we had two patients with Down syndrome, one that had Alzheimer's in addition to Down syndrome, and also another one that was resistant to beta myeloid. So this is again within the pluripotent and unipotent cells also in the case of gliomas, and this is a cooperation that we're doing, collaboration with Dr. Rabinovi and the Tel Aviv University. This is gliomas and brain tumors, and this is a model. We have myocardiocytes, and you can see that when they put together an organoid, you can see how it is beating, because at some point when the 3D dimension overcomes the 2D dimension and it becomes 3D, at some point they start to beat in a synchronous manner, expressing here the phenotype situation, and we can also study some congenital diseases that impact on the heart. This is a good organoid model, and we have the same with the brain organoids. Here you can see a rudimentary cortex, and when we have the culture of the cells, we have a 3D model of the cells, and this model seems to be more efficient because the communications and signals seem to be better. So the brain organoids, heart organoids, are very good models of some congenital diseases that share the hereditary mutation, and FDA is currently mutating towards the use of heart organoids, and they're leaving aside some of the animal models. In the case of Alzheimer, all of the models that are based on mice are not only for Alzheimer, but also hereditary diseases. But the FDA is now using organoid models based on humans to study the toxicity of several drugs. Some of the documents that I recommend as very interesting from the International Association of the Stem Cell Research. There is a periodically updated guideline, and also for the clinical translation of the stem cells, and also the Alliance for Regenerative Medicine. This is an optimistic publication that refers to the clinical trials in Asia Pacific, Europe, North America, and apparently there is a very strong movement also based on the gene therapy, as explained before, but a very important commercial movement that indeed will take place at some point. And here we have a synthesis and applied in approved application. So, for example, we have the standard of stem cells in hematological neoplasias. And here we have, for example, high inflammation situation in the heart that could improve and benefit from the frontier application. So we need to complement biomarkers with the application so that we can segment the medical subentity that will improve with the application of stem cells. And this is clearly Parkinson's disease and also blood issues and all of the stem cell transplants within the central system go beyond the survival of the cells, but also what we know as the wiring, the possibility to be included electrically in the electrically correct manner to the pre-existing brain. We have the first therapy with tumoral infiltrating substances, and we are always referring to these irregular application complexes for stem cells that do not bear in mind the good lab practices, the good practices for clinical trials, and the good manufacturing practices, which would be the safety triangle. And I am showing you stem cells in culture. And the conclusions are that this is a fascinating biological discovery. It is revolutionary. It has an enormous impact. It already had it on the media, and also as to the therapeutic impact, it is to be seen. There is a great evolution in the pipeline. In the coming years, we might see an important therapeutic approach, but for now, this is still under treatment, and for us, it is an important way to create a model and to show these pathologies. Thank you very much. Thank you very much, Gustavo. It is very important to bear in mind the great potential that these technologies have to improve the well-being and the health of individuals. And after listening to the researcher, we would like to look at the sponsor and their perspective. And that's the reason why we have invited Pablo Casarat, the lab from Argentina that is working on the development of an advanced therapy in Argentina. And they're also working with various biotechnological companies in the US. And now we have also our science biotherapeutics that are working on the specialized development and manufacturing of customized interleukins. We have Julio Vega, who is the manager of innovation for the Pablo Casara lab in Argentina, and also in charge of the pharmaceutical development with our science biotherapeutics, and later on we'll have one of his colleagues. But Julio, please go ahead, and thank you for being with us.
Hola, ¿qué tal?
Hello,
Buenas tardes y
good
buenos días.
afternoon, good morning.
Voy a compartir con ustedes
I am going to
esta
share with you this
presentación.
presentation.
Un segundo que la pongo en
Just a
pantalla.
second.
Bueno, se ve bien, me parece, ¿no?
I think that
¿Se
you can
escucha y se
see
ve?
it and hear it properly. Yes, very well.
Entonces, hoy les voy a hablar de
Today I
desafíos
will be
que nosotros
discussing the
encontramos en el
challenges that
desarrollo de
we find
medicamentos
in
de
the
terapia
development
avanzada.
of advanced
Yo soy
therapy
Julio Vega, gerente de
medicines. My
innovación
name
del
is Julio
laboratorio
Vega. I
Pablo
am the
Casará
innovation
en
manager
Buenos
with the
Aires, Argentina.
Pablo Casara lab in Buenos Aires,
Bueno,
Argentina.
nosotros hemos desarrollado dos medicamentos de terapia
We
celular.
have
Uno
developed
es
two
Vaxinel, que es
cell-based
una inmunoterapia
immunotherapy
celular
medicines. One
alogénica
is Vaccinal, that is
para
an
melanoma.
allogenec
Se trata de una
cell-based
suspensión de
immunotherapy
4
for
líneas
melanoma.
tumorales
It has the suspension of
inactivadas
4
por una
tumor
baja dosis de
lines that are
radiación que está
inactivated with a
autorizado
low
bajo condiciones
dose of radiation
especiales en
authorized
Argentina.
under
Y
specific
Neoderm
conditions in
está en proceso
Argentina,
de
and
registro,
Neoderm that
que
is
es un
in the
medicamento
process of
combinado
registering, and
de
it is a
ingeniería de
combined
tejidos para
medicine
reparar
based
úlceras
on tissue
crónicas.
engineering for
Se
the
denomina combinado porque
treatment of
incluye
chronic
una
ulcers. It
membrana
is
de
considered
colágeno
combined
que
because it does
está
include a
ya
collagen
autorizada
membrane that
en la
has already
Argentina
been
como
authorized in
dispositivo
Argentina
médico. A
as
esa
a
membrana nosotros
medical or
sembramos queratinocitos y
device. We
fibroblastos
dot it
austrólogos
with
en la cara superior e
keratinocytes
inferior que Los antes autólogos,
fibroblasts.
provienen
They
del
are
mismo paciente
autologous
y
and
se
they come
amplifican
allogenic and they come from the
en
same
cultivo
patient and
en
they
nuestra planta autorizada ya
are
habilitada por ANMAT.
grown in our plant that has already been authorized by
Bueno, ¿de qué
UNMAT.
desafíos les voy a hablar?
What
Yo
are the
voy a tomar una
challenges?
perspectiva un poco diferente.
I
Donde
will
voy a
address a
hablar de los
different
desafíos
point of view.
del
I will be
desarrollo,
discussing the
empezando por
challenges that we
estandarizar
have with
y
the
diseñar los
development,
procedimientos,
beginning with the
habilitar
standardization
la
and design
planta,
of the
la
procedures, the
medición de potencia, que es algo muy importante dentro del
certification of
desarrollo
the plant,
farmacéutico de estos
the
medicamentos, y
potency
también
measurement
de un
that is very
episodio que si
important with
bien
these
era propio
medicines and something that
de
was
nuestro
typical
propio desarrollo,
of our
me
own
parece
development,
que
but
es
I
una
think that
experiencia
it
que
is an
ayuda a
experience
compartir
that helps
desde
us
ahí.
share as a
Bueno, lo
comparison.
primero
So,
es,
first,
tenemos que compatibilizar
we
un
need
estándar industrial
to
farmacéutico
reconcile
con
an
células
industrial
vivas. Algo
pharmaceutical standard
parecido a
with
lo que el
live
doctor
cells,
Timón
and this is something
explicó. Es
similar
algo
to what was
relativamente
explained by
complejo
Dr.
porque
Timon. It is
las células
something
vivas, por un
complex
lado,
because the
todo lo que es
live cells, on the one
todo lo
hand,
que tiene que ver
everything
con células, pertenece al mundo
related
o
to
de la
cells, it
investigación
belongs
académica
to
muchas
research,
veces,
academic
o de
research
la medicina,
oftentimes,
de
or
la donación y
medicine,
aplicación de
the
órganos,
donation, organ
trasplantes.
donation, and
Entonces, estos
transplants.
dos mundos
So
no
these
tan
two
fáciles de
worlds are not so
compatibilizar.
easy
Es
to
decir, hay que llegar
reconcile. So we need to get
a un
to
diseño en el que las células, digamos, se sientan cómodas dentro del esquema
the
del desarrollo
design where the
farmacéutico.
cells are comfortable as
¿Y
part of this
cómo se
pharmaceutical
puede lograr
design, and
esto?
how
¿O
can
cómo
we attain
nosotros
this, or
lo hemos
how
podido ir
were
logrando?
we
Por un
able
lado,
to
de
attain
parte de la
this? On the
industria, que
one
somos
hand,
nosotros,
from the industry,
es muy
that is
importante tener un
us,
equipo
it is
calificado
very important to
multidisciplinario.
have a
Ese equipo,
multidisciplinary and qualified
en
team.
nuestro caso, involucra químicos, farmacéuticos,
And in that
biólogos,
case, we have
médicos,
chemicals,
gente que está
chemistries,
muy
doctors,
enfocada
persons who are
en todo
very
lo que son asuntos
involved
regulatorios
in
y
everything that
ciencia
has to do with
reguladora. Es
regulations,
muy
and
importante el
it is very
enfoque
important to have
multidisciplinario
this
porque si
multidisciplinary
bien todos los
approach.
medicamentos son multifacéticos, estos
Because even
son
all medicines
particularmente
are
complejos, como
multi-phase.
también
These are
explicó el doctor
particularly
Timón.
complex, as Dr. Timon also
Un
explained.
punto muy importante es la
Something
aplicación
very
de
important
calidades del
is
diseño, es decir, de
the
ciencia y
design
análisis de
quality
riesgo. Un
application.
instrumento que es muy interesante,
Science
muy
and risk
importante
analysis
de
are very
incorporar
important tool
temprano
to
en el
be
diseño del producto
incorporated
y del
early in the
proceso
design
es
and
el
process of
análisis
the
de
product.
riesgo
Is the
que
risk
propone la agencia
analysis
europea
proposed by
en una
the
de sus
European
regulaciones.
agency in
La
one
agencia
of its
europea tiene
regulations
un análisis de riesgo,
where
o propone
there
un
is a
análisis de riesgo
risk
que a nosotros nos
analysis
ha resultado muy
that
útil,
has
sobre
been
todo en el
very
caso de
useful for
Neoderm,
us,
que es el
especially in the
producto
case
más
of
reciente.
Nioderm, our most
La
recent product.
interacción con el sistema
The
científico es muy
interaction with the
importante.
science
Buena
system is
parte
very important.
de la investigación básica e
Most of
incluso
the basic
aplicada
research and
proviene del
also
sistema
applied research
científico,
comes
y también
from
el
the
sistema científico
scientific
muchas veces
system,
puede proveer
which
de
also
soluciones
oftentimes
innovadoras, por
provides
ejemplo, para la determinación de
innovative
potencia.
solutions, for
Y la
example, for the
interacción con la agencia
potency
reguladora, en nuestro caso
determination,
con
and
ANMAT.
there's an
Eso en nuestro
interaction
caso
with
fue
the
muy
regulatory
importante.
agency, in our case,
El
ANMAT, which was
acompañamiento de
very
ANMAT ha
important.
sido muy
ANMAT's
importante.
support has been
Dentro de
very
los
important.
reguladores, a nosotros nos
In the
permitió tener guías
regulatory area,
regulatorias específicas.
we
ANMAT
found
adoptó, por un lado,
having specific
muchas
regulatory
definiciones
guides very
similares
useful,
a las de
and
la
MAT
agencia europea.
adopted
Tiene una guía de estudios
regulations that are
específicos
very
para
similar
terapias,
to those of the
para terapias
European
avanzadas.
agency, especially for advanced
También,
therapies,
como
and they also have
estamos dentro de las
specific
TICs,
GMPs,
tenemos una
and
GMP
within
específica
the
para terapias avanzadas.
case of
Pero más allá
PICS,
de
we
todo
have
eso,
specific GMPs
la
for advanced
asesoría
therapies, but beyond that,
científico-regulatoria de ANMAT, el
ANMAT's
acompañamiento desde etapas bastante tempranas,
scientific
sobre
and
todo en el caso
regulatory
de
support
Neoderm,
since the
fue muy
beginning,
importante
especially since
para
for
detectar
Neoderm,
distintas
was very
brechas
important to
que
detect
tuvimos que
different
ir
gaps
completando
that we
para
had
poder
to be
armar un
bridging
CTD, un Common Technical Document, a la altura
to
de lo
develop
que
a common
ANMAT
technical
requiere.
document up
Bueno,
to UNMAT
Vaccinel
standards.
es básicamente
Vaccinel,
es el proceso. El proceso
here
tiene
I described the
una
process
definición de ingredientes
basically,
activos,
which
que
has
son
a
las cuatro
definition of
líneas
active
celulares,
ingredient, the 4 cell lines that have a quality control. Then they are irradiated. We develop a suspension mixing the 4 lines. This is irradiated, and the irradiated and fraction lines in cryovials are cryopreserved in a liquid nitrogen tank, and then the quality is controlled to condition them and transport these samples in dry ice in the cell lines or refrigerated in the case of the rest of the devices. All of this is necessary, what is needed to apply the immunotherapy. In the case of Nioderm, we said that this was an autologous medicine. We take a skin sample from the patient, which is transported under controlled conditions, and at the facility we separate the dermis from the epidermis, and then we do a culture of fibroblasts and of epidermis keratinocytes, which are the active ingredients that are controlled with a specific protocol. Then this is cultured in the collagen membrane, which, as I said, had already been authorized by ANMAT as a medical device per se. This is controlled, and finally it is conditioned for transportation, and it's administered to cure chronic ulcers. In addition to standardizing the procedure, something very important is to certify the facility with specific rules, in our case, PICS rules. This is a very important table here in the Annex 2A. In good manufacturing practices of PICS with 2 areas that will be guided by GMPs. I'm talking about processing, regulated by ANMAT in Argentina, and there's the donation area and the application of the administration of the medicine. This is regulated by INCUCAI, the agency that regulates all that has to do with donation, transplant, and administration of donated tissues. It's very important to have a certified facility to be able to ensure good compliance with GMPs. Now, measuring potency is also a challenge because it's not the same as to the measure the potency of medicines based on molecules or chemical properties. Here, this is based on cells, so we need to measure the amount of cells and the potential to exert their biological action. In our case, ANMAT's was very important, as I already said, because it helped us to standardize the method to be up to regulatory standards. We adopted a method that distinguished live and not live cells, and also allows us not only that, but also to view the morphology through microscope to see whether they are not only viable, but also apt or conducive to the reparatory action. And a brief experience we had in the selection of comparators for Vaccimel. As I said, Vaccimel is a cell therapy for melanoma. When the clinical study was done, there was no comparator that didn't have enormous adverse effects for this relatively advanced melanoma. It was very difficult to recruit participants for the test because the randomization made it possible for individuals to receive the other authorized treatment, which really had many adverse side effects. So in conclusion, we had to see how to choose comparators. In future, we have not had such an example so far, but the selection of a comparator is critical, not to delay excessively the trial, which would be to the detriment of the benefits for patients, the company, and the regulatory agency. This was a snapshot of our experiences that I thought was good to share with you today. The conclusions: it's a challenge to apply standards to live cells, but this is possible through a specific and clear regulations, including GMPs, and also an early regulatory support, which is very important. And we also need a highly qualified multidiscipline team at the company. This multidisciplinary area is a critical aspect. It's very important that the team would not see just a few aspects, but a whole vision, because sometimes something seems convenient or possible from one point of view, it's prohibitive from another point of view. So thank you now very much. We have accepted at Casara this challenge, and now we will also see if we can work with genetically modified cells. Thank you very much, and thanks again very much for the invitation by PAHO. Thank you very much, Julio. Really, it's fascinating to see this kind of developments in other regions, and we appreciate you having shared these challenges with us. Now, continuing with a sponsor's perspective, we will have Dr. Claudia Zilberberg, who chairs the board of Our Science Biotherapeutics. Claudia, the floor is yours. Can you see me? Thank you very much for the invitation. I will share my screen. Can you see my screen? You need to put it in presentation mode, please.
Okay, bueno, muchísimas gracias. I will speak in English.
Thank you very much.
Si bien although I am from Argentina originally, I've been here in the United States for the last 35 plus years and working in the CGT space. So that is my vocabulary and my lingo. So I'd rather give this presentation in English. As I said, I am part of board member and founder of a couple of companies in this space. Akron Bio is the ancillary material provider for many CAR-T companies today. Our Science Bio is our provider of adjuvants for some cell therapies and custom digital platforms for AI and manufacturing of advanced therapies. In addition to that, I want to make sure that you guys know that I am part of several organizations in the space, and one of them that I want to highlight is the Standard Coordinating Body. It's a body that was founded about a few years ago and produces a lot of standards for this industry by several SDOs like ISO and others. And the upcoming National Academy's workshop on AI and advanced therapies here in November, which I highly recommend for all of you to assist. So everybody talk about cell therapies. I know that Gustavo did a very good run about them. I just want to give some examples so we can relate. Cell therapies could be the CAR-Ts, dendritic cell vaccines, TILs, or NKs. On the gene therapy side, as I said, is AAV replacement of hemophilia and others, or CRISPR edited cells. Tissue engineering like Neoderm, like Julio mentioned a little bit about, and the RNA nucleic-based technologies that we all know about, vaccines like COVID vaccines, oligos, and others. And I wanted to give another perspective from the manufacturing setting. Why this, and I was looking at the questions, why these therapies are so expensive. And I think that basically the models that we have nowadays in terms of how to deliver, how to manufacture these therapies are becoming a little bit cumbersome. This is basically a scheme of how things work nowadays. The hospital or a medical center collects the blood in an apheresis and is transported to a CDMO that does either the cell separation that occurs in the apheresis center or occurs in the CDMO, the activation, the expansion of the cells, the formulation, and shipping to the hospital back for the patients to be infused. So this is in a nutshell what occurs, all the steps that occurs in a high level overview of a CAR-T today in globally and around the world and in the US. On the allogeneic side, we collect cells from a couple of characterized individuals, and those cells are characterized and expanded and create a cell bank and pick up those cells when they are needed. They are cryopreserved, send it to a CDMO. So there is a logistic step involved, create an expansion with media, cytokines, and supplement, which we call ancillary materials. And once that is being achieved, the cells are being banked again and perhaps shipped to different centers and hospitals where the patients are being infused. And I'm going to concentrate on those two examples for now. So the big bucket items are the patient acquisition, the biological sample, the manufacturing, the release, the patient infusion, and what we call today the real world evidence data, the post-infusion patient data that creates a loop back to the developers to create more value into their products. And of course, in each step, because of the location of each one of these activities, there are logistic steps into the equation. All of it in the structure that we work today creates a lot of cost and expenses that makes these therapies quite expensive and quite difficult to be paid by the payers, at least here in the US. So if we go one by one, I will say that the pain points of the biological acquisition material create a pain point on the sample management, on the testing of the material, inventory management, and the logistics, and the patient characterization. Create a patient profile where they are actually able to receive that therapy because it's exactly what they need as a treatment instead of a chemo or another immunotherapy. What are from the menu of therapies in the oncology menu that exists, which one this patient it's able to create perhaps a cure after the treatment with that particular treatment. So those things happen in hospitals, medical collection centers, and sometimes in decentralized point of care, physicians, central medical offices. There is a lot of technologies that need to go attached to it and infrastructure in the digital side that has to do with softwares, ERP systems, inventory management, and logistic software. The pain points on the manufacturing, besides that we are all working on closed systems, so we can eliminate the expense of having a lot of layers of quality compliance on facilities, call them CDMOs, is that create that value that we actually, if we look a little bit back, was created with cord blood banking, and some of the systems that were created at that time for close processing cord blood banking and in the cord blood banking industry, which we believe it's one of the predicates of perhaps what the future may look like. Closed system and be able to work them in environments that don't require a heavy load of regulatory and quality compliance. So the continuous real-time data is something that we are achieving nowadays with novel technologies in the space, and be able to, with that data, create prediction and proactive corrective action. So all the AI world and data that we are seeing and living nowadays can create a lot of value and alleviate these pain points that will create a more efficient way to manufacture these therapies and achieve the efficacy and the compliance in safety that we require. This manufacturing usually happens in CDMOs, but we are all talking about decentralized manufacturing and including hospital settings where this can happen. So the hybrid model perhaps is something that we are all talking about, where the reducing of the costs can be significant, and decentralized points of service. Each manufacturing step requires software, and we know there exists many choices out there. Although some places the data collection is still manual, the volume is not high enough. But there are many opportunities for digital twins, machine learning, and AI for integrations with hospital settings. Again, the release of these therapies is also a pain point. You know, we need to have quality control for the release, distribution, and the logistics. If it's done in a CDMO or satellite place, that cold chain logistic part is quite costly and quite complicated and complex. There are quite a few interesting standards in the SCB promoted by the PDA and ISO that I encourage all of you to have a look and read. The standard coordinating body has a huge menu of standards for this particular space that are really very, very useful. I participated in the ancillary material ones, and actually 2 days ago was just released the best practices by ISSCR for pluripotent stem cells as well. So have a look at all these standardizations that occur in the marketplace, because it's actually very useful for this, for encouraging the right activities in each of your countries. Again, the release can happen in the hospitals, in the CDMOs, and currently a lot of technologies are in place. Patient infusion, I was just pointed out that the data for post-infusion is quite important as we move along. And last but not least, this is a whole list of different pain points that I mentioned before. And I think that we all need to work on them for reducing costs and taking the opportunity to bring technology to make things easier and more accessible. And thank you very much to all of you and to the PAHO for giving me the opportunity to present.
Thank you so much. It seems like the cost challenges are truly among the main worries of many of our participants. Thank you, Claudia. And I'm happy to introduce now to Dr. Peter Marks. So he'll be sharing his reflections on the challenges in developing advanced therapies on his personal capacity, though he's the Senior Vice President for Molecule Development and the Head of Infectious Diseases at Eli Lilly Company in the US. He's a member of National Academy of Medicine, but most of you probably know him because he has been, because of his role at FDA. He joined the FDA in 2012 and served as director of the Center for Biologics Evaluation and Research from 2016 to 2025. Thank you for being with us, Peter.
No, thank you very much. And you can see the slides okay?
Perfectly.
Okay, and I apologize if the internet is a little slow. There's an internet outage here, and I'm on a backup wireless network. So thank you for putting up with that. And thanks to the previous presenters for setting up what I'd like to talk about in about, I'll try to speak for only 10 minutes so we have plenty of time for questions. Currently, there really are such a wide variety of advanced therapies that potentially can bring benefit to people. And there are many challenges. You've already heard about them, everything from their characterization, their regulatory status, up through their cost. But there's been tremendous maturation and growth over the past years of everything from chimeric antigen receptor T cells, genetically modified stem cells, adeno-associated viral gene therapies, and more recently, in vivo genome editing approaches, which may really transform how we address rare genetic diseases, cancers with in vivo CAR T cells, and even inflammatory diseases with in vivo CAR T cells. The hope here is that with the advancement of technology and the ability to move towards uh, approaches where the product, the gene therapy product, is made in the person as opposed to having to be made outside of a person, that the costs could come down significantly and the simplicity of delivering the therapies could come down significantly. We're not quite there yet on these in vivo approaches. There's still some more work to be done, but I think there's a lot of promise here. Um, in the United States, there are a lot of tools available to drug developers that can help in this process, ranging from uh, things that reduce the cost of development, such as orphan drug designation, and that can potentially uh, uh, for instance, not require a fee for the application submission. That's what orphan drug designation does. And then there are other things um, that have been put in place to help speed through products through development. And some of them also help financially for product developers at the end of the process. There is the priority review voucher program, which is currently pending renewal in the United States. That actually, those priority review vouchers, which were given for instance, a rare pediatric disease, when an approval took place, could be then sold by the company that received them for tens of millions of dollars, and that was um, a valuable tool to help drive development in this area. Whether that will be renewed or not, I don't know. But there are some key issues for advanced therapies that we can talk about. And I think no matter how much you want to focus on the clinical, as you've heard through this webinar, manufacturing quality is often central to product safety and effectiveness or efficacy. And I think one of the things we've learned over time is that time spent early on developing processes for product manufacturing and characterization are generally very well spent. That's time well spent, because if you have a product that makes it through development, but then you can't make it consistently, you may not be able to ultimately bring it to the benefit of patients. Now, one of the issues that often comes up early on in development is it's hard sometimes to figure out how to characterize cellular therapy products in particular. And perfect is the enemy of good in product characterization. Sometimes you can be lucky enough that there is a potency assay which is very easy to find, which is reflective of the product activity. Sometimes, though, you're not as lucky, in which case it's better to just follow some product characteristics that you can define and just follow them over time so that you at least have the best sense that you're making the same product over time. That ends up being clinically relevant because we've seen products that have lost clinical effectiveness because there were changes in manufacturing. Sometimes it wasn't until after the clinical loss of efficacy was seen that somebody had to go back and figure out that there was a change in manufacturing that led to that. So really, failure to develop a potency assay or critical quality attributes has resulted in issues later in the clinic if you don't have reproducibility. That said, clinical development is also very important. And when you're developing therapies for small populations, I think very careful planning of the clinical trial program is essential. I think understanding the natural history of disease as much as possible before moving into a clinical trial is important. Understanding whether biomarkers can be helpful that may either biomarkers that may directly or indirectly indicate clinical benefit can sometimes really speed up development, because a biomarker may be able to allow one to get in the United States an accelerated approval or other types of faster approvals rather than waiting for a clinical endpoint. And that can make the difference in some cases between a product becoming available to help people years sooner, particularly in the area of neurologic disease, where sometimes showing that you're stopping a decline in a clinical endpoint can take a few years. Biomarkers can be very helpful. For instance, we've seen that with some of the lysosomal storage disorders. And other things that can be helpful are avoiding needlessly using patients for dose finding. And what I mean by that is that when you're giving, for instance, gene therapy, once you've given an adeno-associated viral gene therapy to someone, generally they're not going to be able to get another dose. That's probably going to change in the future, but right now that's the state of the art. And so generally it's nicest not to have to use an excessive number of people for dose finding. So Bayesian statistical designs in early phase trials to find the right dose of the gene therapy can be helpful, and can be helpful throughout development. Additionally, it's sometimes helpful for these small populations to just consider seamless phase 1, 2, 3 trial designs. In other words, moving directly from dose finding into a pivotal trial, because oftentimes you're only looking at 1 or 2 doses in these kinds of settings anyway. The other piece, though, that I think kind of circles back to what was just said before I started, is that for small patient populations, the financing of these therapies is really critical. And obtaining adequate institutional, government, or philanthropic support in advance to proceed through development really is critical. We've seen too many products fall out of development and patients be essentially stranded in the United States. And so I think we want to, as a community, try to avoid that if at all possible. And it is true, it causes a tremendous amount of distress to the patients and their families and their friends when development programs have been abandoned. And so really, I think at this point in time, it's gotten to the point where, at least if I were developing a rare disease therapy in the United States and I wasn't associated with a large company, I would probably, even as a philanthropic organization, have a backup strategy to advance the product if commercial sponsors fall out. And as much as I wish that weren't the case, what I can say is that in the past year to 2 years, we've lost 50 to 100 companies that have had, single products in development for rare conditions. So having backup strategies may turn out to be important. An incredibly exciting area, I think, just to finish up, but there are still a fair number of challenges. And I think this webinar has done a really nice job of talking about the challenges from actually how you make these products to how you clinically develop them to how you pay for them. So I'll stop there and look forward to questions.
Thank you so much, Peter. That was great. I'm going to transition now to Spanish. Creo que si algo
believe that
que me parece fundamental
if there is something that is
esto
key to mention, it is the importance of investing time to understand the clinical development of the disease. I think that many of you have asked questions throughout this session on some of the ethical aspects, and just to underscore, even though we have not focused on the ethical aspects, to underscore the ethical impact of some of these assertions, please bear in mind that if we are not aware of the development of the disease, oftentimes the therapies for orphan diseases or rare diseases will mean that it is difficult to assess in a clinical trial or in an experiment what is to be attributed to the disease and its natural development and what is to be attributed to the intervention that is being studied. Now, we have a moment to have a dialogue with our panel members, and this will be coordinated by Franca Carracedo, who is also from the Bioethics department, Sara Carracedo. And in addition to the panel members who have also participated, we will have, as part of this session, Patricia Saidon, Dr. Saidon, who is our expert on clinical trials within quality and regulation of medicines and health technologies at PAHO. So, panel members, colleagues,
turn your cameras on for this conversation that will be led by Sarah.
Gracias, Carla, por la presentación.
I thank you, Carla, for the presentation. I also thank all of the panel members for their presentations. I believe that you can turn on your cameras so that we can all see each other. And there are several questions, from general issues to more specific issues, and I believe that if something is clear, is that advanced therapies need to be safe and efficient to be helpful to the population, and that's why it is so important to have research. Along those lines, there are several references to the regulatory frameworks in the region, and I would probably like to ask whether in Spain, for example, the clinical trials rule is sufficient to be used with advanced therapies, or do you have more specific rules? And this question is clearly for Marcos. I thank you, and I hope you can hear me. Yes. We have the regulations for clinical trials that can be perfectly implemented and used with advanced therapies, and in Spain we have a long tradition with advanced therapies. We have been leading the number of clinical trials with advanced therapies within the European Union for many years, and this continues to be the case. There is a great deal of activities within our organization, our agency, to support innovation, which is the entry point for the academic researchers or small and large companies that would like to develop their clinical research in Spain so that they can clearly work with us. But the regulatory framework is the same one that we use for all the clinical trials, and it works very well. As to evaluation and the technical aspect, we do need some adaptation, but to that end, we have the ones who are in charge of assessments so that we can adapt the regulations to the specific characteristics of these therapies. We know that they are highly specialized. We know that they have special manufacturing processes, but we do comply with the regulations in force. Thank you. Patricia, this is a question to you to understand the regulatory framework on clinical trials and clinical trials with advanced
mencionarnos algo?
therapies in the region. Could you please share something with us? I agree with Marcos. Clinical trial guidelines are based on international standards for good clinical practices, and they are sufficient to assess the advanced therapies, and the evaluators, the ones in charge of assessment, need to be trained for that assessment and follow up. And that's the reason why in our region we need to have specific training for the regulators so that we can work together with the various areas. For those who look into the registries and also the protocols. And I wanted to share with you that there are some standards for the registry of these advanced therapies, and this is very important when we need to assess these therapies so that progress is made for the regulatory framework and so that all of the countries can have regulations for advanced therapies. And I would like to go back to what Julio mentioned and what Marcos mentioned, the need to have the regulatory agency work with the regulators so that there is a clear expression of what they deem necessary through the development and the importance of that collaboration. It is also useful to have training for the ethics committees that sometimes have to review these protocols. And here I leave an initiative with you, Sarah, so that it is later on discussed as to whether experts are invited to participate, whether these are specific committees or some national, regional committees, that is to say, different committees at different levels. But we all need to be trained, and that's why I celebrate this seminar.
Gracias, Patricia. Y justo ese
I
tocaba el
thank you,
tema
Patricia.
que quería
And this
traer
also
Julio
leads us
a más
to
detalle, que era uno
something
de,
that
de
I
los
wanted
desafíos
Julio
y bueno, una de las
to
conclusiones que
elaborate on,
manifestaba el
and it
seminario,
is
¿no?
one
Un
of the
acompañamiento
challenges that he
regulatorio
mentioned at the end.
temprano. En tu
Early
experiencia que
regulatory
tienes, ¿cómo ha sido ese
collaboration.
acompañamiento regulatorio temprano?
In
¿Cómo
your
debería ser?
experience,
¿Qué es lo que
how
deberían
should
tener en
this
cuenta las
collaboration
ARNs,
take
las
place?
autoridades
What should
regulatorias,
the
al
regulatory
momento de
authorities
evaluar, ¿no?
take
De
into
saber
account
que
at the
hay
time
o que
when
van
they
a
need to
someterse a la
assess
autorización ensayos clínicos sobre terapias avanzadas?
medi
Bueno,
advanced
yo
therapy
para
trials
mí el
when they
acompañamiento
need to
fue muy
submit
importante
those
incluso en el
therapies for
desarrollo no solo
authorization?
clínico, sino
Well, in my
farmacéutico.
opinion, it is
Y
important
es
to
importante
work from
porque
the clinical and
la
pharmaceutical
agencia
point of view.
reguladora tiene una
We
visión
need to
un poco
collaborate,
diferente,
and
más
the
que nosotros
regulatory
tenemos equipos
agency does
multidisciplinarios.
have a
La
different
agencia lo ve
approach.
y
Even
lo ve
though
desde
we
otra
have
perspectiva.
interdisciplinary
En
teams, the
nuestro
agency
caso,
looks
ANMAT
at this differently. In our case,
fue,
ANMAT
estuvo muy disponible para
was highly
dialogar con mucha
available
transparencia.
to
Diría
have
que
a
fuimos muy
very
transparentes.
transparent
La transparencia es muy importante.
dialogue.
También es importante la
Transparency is
humildad,
very
estar seguro de que
important,
ninguno
as well
sabe
as
todo y de
humility,
que
just to be
entre los
certain that
dos
not
se puede llegar a algo
no one
mejor que
knows
cualquiera de las
everything,
dos partes por separado.
and
Y
two parties may get to something
por otro
better
lado,
than
encontramos también
two
en
parties
ANMAT
together may
gente
get to something
muy
better than
informada
two
sobre el
separate
tema,
parties. And we
muy
also
preparada y
think that
con la mente
ANMAT
muy
is well
abierta.
prepared,
El caso
has
del
a
director de
great deal of
biológicos, por ejemplo,
information. For
mente
example,
sumamente
the director
abierta,
of biologics
entendiendo que,
has
bueno,
a
que estos
very open
medicamentos
mind, and with the
son
understanding
un
that
poco
these
diferentes a
medicines
otros
are a little
y
bit
que
different
requieren
from others, and that they do
de
require
conceptos, yo no sé si decir más flexibles,
more
pero de alguna
flexible
manera más
concepts,
flexibles
or
y
in a
sobre
way
todo
more
adaptados,
flexible
digamos, al
concepts,
ingrediente activo que
and they
son
need to
células
be
vivas,
adapted to
que
the
es muy
live
diferente
ingredient,
a
that is
una entidad química, una
live
molécula.
cells, which is
Así que
different from a chemical
yo creo
entity such
eso.
as a
Y en cuanto
molecule.
a un ensayo
So,
clínico en sí, ya les dije, para
as to
nosotros
a
lo más
clinical
complejo
trial as
fue el
such,
comparador,
for us,
más
the
que nuestro
comparator,
producto,
was
Pero
the
bueno,
most
esto creo que
difficult
depende
aspect,
mucho
but I think that
de
this is
cada
closely related
medicamento, puede ser
to
muy variado.
the
Nosotros no
medicines.
tuvimos nada que ver
Each
con terapia
medicine may
génica, por
be
ejemplo. En
very
Argentina hay
varied.
una
We had
resolución
nothing to
ministerial
do
que es
with
bastante
gene
cuidadosa
therapy.
con
There
ese
is a
tipo de
ministerial
terapias,
resolution in Argentina
no
that
solo
is
génicas,
quite
sino celulares, cuando
thorough in terms of that, not
en
only
algunos casos,
gene
digamos,
therapy, but
particulares,
also molecular
nosotros no hemos tenido
therapies.
esa
In some
experiencia, ni
cases, we
en
did
nuestro
not
caso ha
have
sido
that
relativamente
experience, and
simple,
our case was quite
pero
simple.
muy importante el rol
But
del
it
médico.
is very important to have
Gracias,
unmapped.
Julio.
Thank you. I
Peter, no
thank
sé
you,
si
Julio. Peter,
tal vez tengas tú
I don't know
alguna
whether
reflexión
you
sobre esto del
have any
acompañamiento
thoughts about
regulatorio
how to
temprano,
collaborate
estrecho,
early
con los
with the regulatory
patrocinadores
aspect with the
al momento de la
sponsors
realización del ensayo clínico.
when implementing the clinical trial.
Yeah, thank you. I mean, this is one of the more challenging areas that I think WHO was trying to work with a variety of other countries on, and I hope that we'll get back to doing so, because particularly for very small populations, having a regulatory plan that will be accepted by multiple countries so that wherever the clinical trial is conducted, the rare disease patients or patients in need can get access to the therapy is something that's very important. And particularly for small patient populations treated by many of the cell and gene therapies today, there's no reason why we shouldn't have that alignment. So hopefully that the work will continue. Unfortunately, over the course of time, just by the nature of different regulatory structures developing, rather than harmonization, there's been just divergence. But for instance, when FDA talked with EMA, some of that divergence, it wasn't it was just unintentional. It was not intentionally different. It was just divergent evolution. So it just means that some convergence efforts may be necessary, but it would make things a lot better, I think, for both the product developers as well as for patients who would get access more rapidly.
Gracias, Peter. Gracias,
I thank you,
Julio.
Peter.
Creo que
Julio,
ahora
I believe
podemos
that
quizás
now
hablar un poco más sobre el
we can
financiamiento,
discuss the funding,
que es una preocupación además
which
que ha
is a
sido tocada por
concern, which has
casi
already been
todas las
mentioned
presentaciones,
in all of the
sobre todo
presentations.
si es que estas terapias
In particular, whether
generalmente son para
these
enfermedades
therapies
raras,
are for
para
rare
enfermedades
diseases
huérfanas, ¿no? En
or
donde
orphan
tal vez la
diseases
inversión es
for
más
which
difícil de
investment
recuperar.
might be
eh,
harder to
y
recover.
justo hace poco, en la
And a
última
short while
sesión del
ago,
Consejo
in
Directivo
the
de
last
la
session of
organización de
the
la OMS,
Directing
se
Council
aprobó
at PAHO, a
una política
policy was
regional para,
approved, a
digamos,
regional
expandir
policy
el
was
acceso
approved to
a
expand access
tecnologías de salud de alto
to
costo.
high
Entonces,
cost
quisiera saber,
health
y es una
technologies.
pregunta abierta para
So
todos
this is an
ustedes,
open
¿qué
question to all of
estrategias,
you.
qué
I would like to know
iniciativas
the
creen que
strategies, the
debemos
initiatives
considerar
that
para
in your
anticipar
opinion we should
este
consider to
acceso a estas
get
tecnologías y para que realmente
access
lleguen
to
a
the
aquellas
technology,
personas
get early
que las
access
necesitan?
so that this gets in the hands of those who need them the most.
Sorry.
Perdón.
No, no.
Adelante.
Una de las formas que I believe the conversation is going is to create platforms, and I think that there is a guidance or there is una directiva dentro de la FDA de crear
There's a directive
plataformas
within
para que
FDA
puedan
to create platforms to
estandarizar partes del
include, to
proceso que
standardize
lo haga
part of the
más
process so that it
repetitivo y lo
is more
puedan llegar
repetitive
a reducir los costos
and
de
costs
esa
can be
manera. Creo que esa es
reduced.
una de las
So I think that
formas de empezar
that is
a
one of the
pensar en la
ways to start
reducción
thinking
de
about
costos y
cost
de vuelta
reduction.
pensar
And once again,
en hybrid models between CDMOs and hospitals, because I believe that there is a very high cost there that needs to be taken into consideration.
Gracias, Claudia. No sé si alguien
I
más.
thank you, Claudia. I don't
mencionar
know
algo
if
sobre
anyone else would like to refer to this
Sí,
topic.
yo quería añadir algunas cosas.
I would
En
like
primer lugar,
to add
evidentemente,
some
habría que
ideas.
preguntarse por
First,
qué
we
los
need to
costes
wonder
son
why the
altos y
costs
uno de
are
los
high.
factores que
And
lo
one of
explica es
the
claramente el cost of goods,
factors explaining this
los
has to
costes
do with the
de
cost of
producción.
goods, the cost of production.
Aquí obviamente hay
Here, clearly,
mucho espacio para
there
la
is
mejora.
still room for
En
improvement.
los tratamientos autólogos, pues se ha explicado
In the case of some
los problemas logísticos de llevar
autologous
el material de
treatment, we
partida hasta el
have, for
lugar,
example,
devolver las células al paciente
the
y esto pues
problem
aquí
of
hay
how the
aproximaciones que
cells
están
are
empezando
moved
a
to different
desarrollar
places and then back to the
en
patient.
muchos sitios,
So
como la
we
fabricación
already have
descentralizada o próxima al paciente que
approaches
podrían
such
ayudar
as
aquí.
the
Pero también en otros
manufacturing
medicamentos
close to
más
the
de
patient.
fabricación más clásica,
And there
digamos,
are
de
some other
terapia génica
classical
con
manufacturing
vectores virales, por ejemplo,
issues
donde
such
no
as
hay
viral
este problema. Sí
vector
que es verdad
production,
que
where we
son
don't have this
productos
issue
novedosos
of
donde la fabricación a escala aún no
transportation.
está
These are
optimizada. En el caso
novelty
de los
products, and some
vectores
have
RNA
optimized
asociados, por
production. In
ejemplo,
the case
que
of the
están muy usados en muchos
associated
protocolos de
adenovirus that
terapia
are
génica,
part of some of
los
the gene
procesos de fabricación ahora
therapies,
mismo son
the
bastante poco eficientes y
manufacturing
generan un
products are
montón
not very
de impurezas
efficient, and there is a high level of
y el
impurities,
rendimiento,
and
digamos, del producto
the
activo es muy bajo. O
performance
sea que
of
aquí hay
the active product is quite low.
mucho
So
espacio
here
para
we
la
have
mejora. Y
a
luego,
great
claro,
deal of room
al
for
final si son
improvement.
medicamentos
And at the end
para
of the day,
enfermedades, pues
if
siempre
these
va a tener
are
un coste mayor porque
medicines for
las
rare
compañías van a
diseases, the
querer
cost is going to be higher
recuperar
because the companies will
la
want to
inversión
recover
rápidamente, ¿no? Y aquí sí,
the
pues
investment.
podríamos empezar a ver diferentes alternativas
And here
a
we
cómo
can have
pagarlos. En
various
España se está explorando, por
alternatives
ejemplo,
as to how to pay for this.
el
In
pago
Spain,
por
for example,
resultado,
we
ya
are
que
looking
son
into
terapias que en muchos casos
result-based
pretenden
payment,
curar al paciente,
and
pero
in
cuando se utilizan,
often
obviamente, no sabemos
cases
si el paciente ha
there is
curado de
an
por vida, porque
idea to cure
tenemos
the patient, but
resultados
once they're
de
authorized,
seguimiento
we don't
todavía
know
cortos
whether the
y esto genera
patient is
incertidumbre que
cured
nos
or not
trasladan
because
luego
we have
a la hora
short-term
de
results
pagar
that
el
need to
producto,
be
¿no? Y la
borne
idea
in
aquí es un
mind at
poco
the time of paying for the
que la
product.
compañía o
And the idea
el
is that
dueño
the
del medicamento
company
también
or the
comparta
owner of
parte
the
de estas
medicine
incertidumbres y vaya cobrando a medida que el
also is part of
paciente
this
vaya
uncertainty and
cumpliendo
collects
ciertos
the
milestones,
money as
ciertos
some
hitos.
milestones are attained by the patient. Thank you.
Gracias, Marcos. No sé si
I
alguien más
thank you,
quisiera agregar
Marcos. I don't know if anyone else would
algo
like to add
sobre este punto.
something on this.
No. Una pregunta que quizás está más ligada
A
al
question
rol
that
del
is
investigador. Tal
probably
vez,
more linked
Gustavo,
to the role of the
quieras decirnos algo.
researcher, and
¿Qué
Gustavo, you might
desafíos en
have something to
específico
say.
se
What are
encuentra
the
uno como
specific
investigador al momento de
challenges that a researcher faces
realizar
at
ensayos
the
clínicos
moment of
sobre terapias
conducting
avanzadas? ¿Cuál ha
clinical
sido,
trials on
digamos,
advanced
la relación con los
therapies?
comités de ética?
What has been
Quizás
the
que ha sido
relationship with the
mencionado
ethics
en
committees?
presentaciones,
And this
en
has
intervenciones
already
previas
been
y también
mentioned
en
in
el chat. ¿Cómo ha
earlier
sido, digamos, ese
presentations and through the
intercambio
chat.
como
How
investigador en el
would you
proceso?
characterize that exchange with the practitioners throughout the process?
Gustavo, creo que estás en
Gustavo,
mute.
I think that you are muted.
Creo que no me está
I don't think
escuchando.
he can hear me.
Pero a ver si no podemos
Otherwise,
continuar con
we
otra
can continue
pregunta que quizás
with
es
another
más
question
rápida mientras damos
that
chance
is
por el
faster.
audio. Había una pregunta sobre
There was a question
los xenotrasplantes.
Ahí
Si es
tengo
que
mi,
es...
on
ahí
the
tengo mi audio, perdón.
Perdón, disculpa.
I
Discúlpame.
turned
No,
on the
no
mic. I
lograba
apologize. I was
desbloquear el
unable
audio. Ahora
to
sí estamos.
activate the mic. We
Creo que hay
are
dos o
fine
tres
now, and I think
comentarios en
that there are
función de
2
lo
or 3
que
comments
recién
based on
decía.
what
Nosotros
you
hicimos
just
un
mentioned.
ensayo
We
clínico
conducted a
inicial
clinical
de la historia de
trial
estas células
based on the
para el
history
accidente
of these
cerebrovascular
cells for
en etapa subaguda,
strokes
intentando
in the
salvar lo que se conoce
subacute
como la parte de
stage, trying
penumbra a
to
partir de células
save
madre
the
no hematopoyéticas, células madres
patients
autólogas,
in the
mesenquimales, no
case of
hematopoyéticas. Y en aquel momento
non-autophagic
tuvimos mucha dificultad con el
mothers, and
reclutamiento.
we had issues with the
Tuvimos todas las
recruiting of the
autorizaciones
patients. We
necesarias,
had
pero el
all of the
reclutamiento
authorizations,
fue
but the
complejo. De hecho, no
recruitment
logramos
was
reclutar la
quite
cantidad de pacientes
complex. We were
suficiente
unable to
como
obtain a
para poder tener
sufficient
resultados
number of
válidos.
patients to
Y de
have
alguna
valid
manera
results.
tuvo que ver
And
con
in a way,
que
this was
me parece que era
also
una tecnología
related
probablemente
to the
anticipada,
fact that it was
tiene que
an
haber una
early
coordinación entre la época y la
technology. There has
tecnología.
to be some agreement
Muchas
between the
veces las
technology
tecnologías
and the
aparecen
times, and
cuando no
sometimes the
tienen cierta
technologies
madurez y es
come up when
difícil
they
la
are not
implementación.
mature
Así que
enough and they are
nuestra
difficult to
experiencia no fue
implement.
buena en el
So our
sentido del
experience was not
reclutamiento
good, meaning
de
recruitment
los pacientes
of
necesarios
the
para llevar a
patients, the
cabo el
necessary
proceso.
patients
Y
to
hoy
carry
yo
out
le
this
diría que en
process. And
Argentina,
today I would
más
tell
allá
you
de la
that
experiencia de
in
Casara, que
Argentina,
es
beyond the
notable y que es muy
Casara
importante, pero
experience, that is
probablemente debe
quite
ser una de las pocas que tenga el desarrollo,
remarkable, it might be one of the few that
digamos, el
has
grado de desarrollo
the
que
level
es necesario
of
como para
development
poder
that
tener
is
resultados
necessary
válidos.
to
Y
have
el otro
valid
aspecto que a mí me parece muy
results.
importante y
And
que creo que
another
se mencionó
aspect that, in
recién
my
es la
opinion, is
aplicación de los
important and that I
biomarcadores.
believe that
O
was
sea, es
recently
algo que me
mentioned
parece
has to do
esencial
with
en
the
el sentido que
application of
nos da
biomarkers. It is
alternativas de
key
medición
because this gives us
importantes en este
alternatives,
momento validadas,
significant
que más
alternatives
allá de
for
la clínica
measuring,
o en
which
enfermedades
are
donde
already
por ahí el
validated. And
efecto,
beyond the
recién
clinical
creo que lo
aspect,
comentaban,
when
el
the
efecto del
effect, the
tratamiento,
effect, the
el
impact of
efecto
treatment,
clínico del
the
tratamiento
clinical
puede demorarse
impact of the
bastante y puede
clinical
haber un
treatment
efecto en
may
los
take a
biomarcadores que hoy por hoy
while,
son una
and there
buena
could be an
alternativa
impact on the
complementaria
biomarkers that now
en la evolución
are
de la enfermedad.
a
Así que yo
good
creo que el
option
desarrollo de biomarcadores
to
específicos para las
based on the
enfermedades
evolution of the disease. So
complementa
I
la
think
evaluación del
that
efecto
the
terapéutico o
follow-up of
no
the
de las
biomarkers
terapias
also
avanzadas.
complements the effect of these advanced therapies,
Gracias,
treatments.
Gustavo.
Thank you,
Y como el
Gustavo.
tiempo se nos acaba y
And
no vamos a
since
poder
we
terminar con todas las
won't be
preguntas
able
del
to
chat,
answer all
quizás
of the
para
questions
una
on the
pregunta a todos
chat,
también
this is
abierta
a question
para
to
ir cerrando
everyone,
esta
an
conversación,
open question to
si tendrían
put an
que
end to this
elegir una o dos
conversation. If you had to
acciones
choose one or
prioritarias
two
para la región
priorities for
para
the
que
region, priority
pueda
actions for the
incentivar, pueda
region
realizar
to
ensayos clínicos sobre
carry
terapias
out
avanzadas,
clinical
¿cuáles,
trials
cuáles
on
serían esas?
advanced therapies, what would be those?
Estás abierta para
This
quien
is open.
quiera
Whoever wants to
contestar primero.
answer.
Sí, yo quiero hacer un comentario a lo que dijo
I'd like
Patricia
to
Saidón.
add a
El
comment to
entrenamiento y la
what
capacitación de
Patricia
los comités
Saidon
de
said. The
ética. Me
training
parece fundamental. El
of
comité de ética
the
es la
ethic
primera
review committees, I
relación que
think that
tiene el
the ethics
investigador para
committee
la
is the first
validación de su
relationship that the
protocolo
researcher
y
has to
muchas veces los
validate the
comités de
protocol,
ética,
and
por lo menos en
oftentimes the
Argentina, son
ethics
heterogéneos en su
committees are
formación y tienen
heterogeneous
que
in Argentina,
lidiar con
and they
muchas cosas
need
diferentes de
to
muchos órganos, sistemas
face
y enfermedades
several
y
different aspects
a veces es
within the
difícil que la
disease
evaluación ética y la
spectrum,
evaluación
and
científica de un
sometimes the
protocolo la
ethical
hagan.
review of a given
Probablemente en
protocol is
este momento lo
difficult.
mejor sea buscar un
Right now, it
asesoramiento
might be a good
externo
idea to
más
have
que comités
external
específicos, pero
advice
cualquiera de
more
las
than
opciones valía
specific
discutir.
committees,
Pero a mí
but
me parece
any
que
of the
el
other options is
fortalecimiento de los
valid to
comités de
discuss. But
ética
I think
para
that
la
strengthening the ethic
comprensión y
committees for the
validación de las
understanding and
terapias avanzadas
validation
es el
of the
primer
advanced
paso, es
therapies is
esencial
the first step.
como
It
para
is
poder
key
empezar
to be able
a validar este tipo de
to
terapias.
validate this type of therapy.
Y la educación del
Yes, the
workforce y la
education of the
educación de la
workforce,
comunidad médica
as well as
estas,
that of the medical
estas oportunidades
community,
de tratamiento
to
son
inform them of
extremadamente
the
importantes. El first step.
treatment opportunities. This would be the first step.
Marcos, Peter, Julio, ¿quieren
Marcos,
agregar
Peter,
algo?
Julio, would you like to add
Solo quería
anything?
añadir, pero
I
hablo de
just
manera
wanted
interesada, creo
to
que
add,
una
and
regulación,
here, of course,
digamos,
I have some
sólida y
self-interest,
clara
but I
que
think that
marque
a solid
estos
organization that
tratamientos dentro de la regulación farmacéutica creo que
clears
es
the path
prioritario. Y
for
luego
this
también,
type of
obviamente, la
therapies within
disponibilidad
the pharmaceutical
de
sector, and
personal
clearly the
capacitado
availability of trained
para su
staff
desarrollo y su evaluación.
for their development and evaluation.
From my perspective, I think the ability to have some kind of regulatory harmonization to make it easier to move across country lines with these therapies so that we don't have to redo everything each time a new country wants to evaluate it, that would be very helpful.
Sí, una cosa más es lo del acompañamiento, eso es
I
muy
would
importante.
add
Reglas
support,
claras, seguro,
clear rules, certainly,
con la flexibilidad
with the right flexibility, very good training for the regulatory agency and the company, with interdisciplinary teams and support. In my opinion, this is key. Without a dialogue, one follows a path that may take us nowhere. And let me add, for the region, the need, as Marcos said, for everyone to have regulatory standards, and also to communicate the need that everything has to be done within the regulatory framework in force, and the need to train evaluators and also the ethics committees and the community in general, as Claudia mentioned, in connection with these therapies.
Muchísimas gracias. Claudia,
Thank
¿quieres
you.
agregar algo?
Claudia, would you
Una
like
sola
to
cosa,
add
these
something? Just one
therapies,
more
at least
thing.
the autologous one, necesitan scale out, necesitan global presence. If you don't have global presence, these therapies are not moving forward, and that's why many companies fall out. They need to scale out, and scale out means that every region in the world needs to access and have the framework to receive them. So bear in mind that this is something that is needed urgently.
Ahora sí, muchísimas gracias a
Now, I
todos
would like to
por la
thank you all
discusión. Carla,
for this discussion.
te doy
Carla,
la palabra
I give you the
para el
floor
cierre.
for
Gracias.
the closing comments. Thank you.
Muchísimas gracias
Thank you very much to our panel members for their presentations, for their contributions to the discussion. I believe that not only the virtual hands we see, but also the comments tell us clearly that our region values your contribution, this dialogue, and I would like to underscore from the point of view of PAHO that we work systematically in an organized fashion, in a consistent fashion, so that all of the countries in the region have solid and efficient mechanisms for ethics reviews and regulatory reviews. We are in diverse regions. Some countries are more advanced, others are just implementing their regulatory agencies, and we need to learn from each other. We do not need to reinvent the wheel. We do have standards for clinical trials. We do have ethical and regulatory standards. We have the experience of other countries, and by adopting these standards, seeing how to harmonize and avoiding duplication and moving forward efficiently, we will move a great deal forward in our region. Thanks again. This session has been recorded. The recording will be sent in Spanish and in English using our ethical review list. It will also be published on the portal of clinical trials for the Americas and also on the Bioethics website. The next session will be on November 4 on vulnerability. This will have to do with the core of research, and you're all invited to this session. I thank again all of the panel members. And we leave you with some QR codes for our participants in this session to be able to have access to many of the documents that we have shared with you. Have a good afternoon or a good end of the day. Thank you.